Next, we are going to discuss the transverse section of Dicotstem. As in the previous lesson, you had an understanding about the various stems and roots of both dicot and monocot plants. There was a video illustration in which you saw what is the arrangement, and because it was an overview, now we are going to see one by one into each stem and each root that what basically it is comprised of, what are the cells, what is the arrangement, and what is the purpose. Okay, for just an in-depth knowledge. Now we are going to consider the dicot stem. It's wrong, it's not dicot stem. We're going to consider dicot root over here. The one I drew is dicot root while I wrote dicot stem. Please don't get confused. This is a dicot root we are talking about, and we are seeing its transverse section. My way of presenting what a dicot root looks like. This is a transverse section. You can just imagine that it would be a circular section because the root would be circular. This is a part of it that we have considered, and the part includes the three parts. Now, what are those three parts? The first one is epiblema. That is the epidermal layer. As you can see over here, this part shows epiblema. I would write the parts over here so that it is clear. This part, this much part where we have little protrusions, the red part, the cells, tubular cells, that is epiblema, or as we had discussed the word epidermis. This part is epiblema. Now the second part of the dicot root would be from here till this red layer that you can see. This red layer, this red part, it is the tubular cells, which are cylindrical sort of cells, which are arranged and they are tightly packed. This layer is known as endodermis. Now we have an epidermis called epiblema, and we have another red part, that is endodermis. Now do keep in mind that from the layer beneath epiblema, that is epidermal layer, beneath epidermis, there starts a layer of cells, many layer of cells, okay, many layers which are having parenchymatous cells. These layers till epidermis, they comprise the region known as cortex, okay, as is told over here. Then from epidermis, inside, going inside, this particular part, excluding the epidermis, this particular part that comprises the vascular bundles is known as stele. Okay, now in the stele, what you see over here is that there is part of cells. This is one color combinations you can see. There are different types of cells. I have used different colors for depicting the different types of cells. There are cells, and we have inside the stele, we have written over here, there is a pericycle, that is again parenchymatous cells. There are vascular bundles which comprise of xylem and phloem, and there is a pith. Now because we are talking about dicot root, do keep in mind that the pith which is present in the center, this particular part is pith. This part is pith. You must be knowing it. This pith is highly reduced in a dicot root. Okay, the pith is almost negligible, very much reduced. Now we have to keep this pith in mind, because basically when we study these structures, what we have to come to, what we have to reach in the end is the basic difference. Anatomical differences would be the points from where you can easily remember what a dicot stem is, what a dicot root is, how it is different from a monocot root, how it is different from a monocot stem. So do keep in mind that the points of differences should be very much clear in your mind. Except anything, the points of differences should be clear, and the first thing that you have to remember is that the pith is highly reduced in a dicot root. Now talking one by one, talking about epiblema, the other name for epiblema is pyliferous layer. Remember it? Now this pyliferous layer is having living tubular cells, as I have mentioned over here. They are living tubular cells. Now there would be parenchymatous cells. Okay, you can remember this from PP. Now you remember what parenchymatous cells were. We had studied parenchymatous cells are circular in shape. They are living cells. They have the living material, storage material inside them. Now these parenchymatous cells in a dicot root, because it is a root and because the purpose of root is absorption of water and minerals from the soil. So in order to accomplish that absorption in a better way, what is required of these cells is that they have large surface area, and for that large surface area, these epiblema cells, they have their outer surface protrusions. As you can see over here, this outer surface protrusion is a root hair. Okay, the epidermal cells, their outer wall has protruded, and when the protrusion goes so long, you can associate it with a balloon. When you have a balloon, as it is not inflated, it is having thick membrane, but when you inflate it with air, the membrane becomes thinner and thinner as more air gets inside. So they are extending their outer surface, so they become thinner and thinner, and at the same time the surface area also increases, which enables a better absorption of root water and minerals by the root. So it is the root hair that is responsible for absorption, and the root hair is the characteristic feature in the case of dicot root, which enable the surface area extension and enable the absorption. One thing you have to remember. Second thing, you have to remember that they are living tubular cells. If they will not be living, if they would be dead, so they will not be able to conduct the water content, okay? So, or rather I would say they would not be able to absorb it. We deal with it in the physiology, but for now you have to remember that the pyliferous layer has root hairs, and those root hairs are extension of these cells only. They are living tubular cells, and this particular layer known as epiblema is single layered. They are made up of parenchymatous cells. Moving further, we have the cortex, which extends from the layer beneath the epiblema up to the innermost layer that surrounds the stele, known as endodermis. Okay, now what do we talk about this cortical layer? Again, they are parenchymatous cells, as I told you. That means this would be a living part of the root. They are green in color, as it has been shown over here. That means they must be performing photosynthesis as well, little bit of photosynthesis, whatever is required, or otherwise they would be responsible for storage. Now, why I am saying or otherwise for storage? Because this is a root that we are talking about. A root would not perform photosynthesis. It would not be subjected to the light. How would it perform photosynthesis? So there is a least chance that there would be presence of chlorophyll in the cells. So we are going to consider that these are parenchymatous cortical cells which are responsible for storage. Okay. Now there are many layers of parenchymatous cells inside the cortex, many circular layers, and one thing you have to remember is that there is lot of intercellular space. Now this intercellular space also has its purpose, that while the water is to be conducted, these intercellular spaces are also a region from where the water is conducted. Apart from the living cells which take up the water, the intercellular spaces which are present, like over here, these are the intercellular spaces. These places would also be responsible for some sort of conduction of water, so keep that in mind as well. Moving further, we have a barrel shaped cell layer, okay, closely packed, in fact tightly packed, so closely that it looks like that many barrels are placed next to each other. This barrel shaped layer is made up of, again, parenchymatous cells, and this layer is known as endodermis. Now in this endodermis, you have to remember that there is a characteristic feature in dicot root, that is the endodermis has Casparian strips. Alright, now what are these Casparian strips? These Casparian strips are the cuticle layer. Subrin or cuticle would be together accumulated at the edges of the cell, which stops the water. This is a waxy material. Subrin coated cells they would be, or the strip would be made up of subrin, and it will not let the water pass through it. Okay, you know, through wax water cannot pass. So we have a Casparian strip that is present in the endodermal cells. Now do keep in mind that this endodermis is devoid of intercellular spaces. It is having Casparian strips. So these Casparian strips has a role when this strip has a role when we talk about conduction of water, but for now you just have to remember the anatomical aspect, that these endodermal cells are closely packed to each other. They are having a suberized Casparian strip, which is a waxy strip, that is cuticle sort of. Till now we have seen that there is nowhere presence of cuticle. Even in the case of Epiblema, there was no cuticle which was present, and in epidermis we are going to find it. Now this epidermis lines the entire stele. Okay, now what this entire stele is all about, the stele has pericycle. Now this pericycle is the layer beneath the endodermis. So the layer beneath the endodermis, which is again made up of parenchymatous cells. Again you have to remember that P for pericycle, pericycle has parenchymatous cells, that is beneath the endodermis. After that, what we get is vascular bundles. Vascular bundles in the case of Dicot root are radial. Now you have to remember these terms. Firstly, they are radial. We had talked about what radial vascular bundles are in the previous class. Again I am going to repeat it, that supposedly this is the stele, such is the arrangement, that xylem and phloem, there are two types of vascular bundles, one is xylem, one is phloem. They are responsible for conduction of water and food respectively in the plants. How they are arranged, we have different arrangements. The vascular bundle arrangement, where both xylem and phloem are differently present on different radii, that particular arrangement is known as radial arrangement, and that we see in the case of roots. Now this is xylem, this is phloem. Over here you are going to find phloem. So you see xylem is on a different radius, phloem is on a different radius. Here also the same thing has taken place. Xylem and phloem are on different radii. So what we see over here is, this is xylem. The blue representation, as you can see, is of xylem, and the black part that I have made over here, that is phloem. In between xylem and phloem there is again parenchymatous tissue which is present below the pericycle, and that is known as conjunctive tissue. Please remember, okay, conjunctive tissue, not conjunctive, it is conjunctive tissue. This conjunctive tissue lies between xylem and phloem. Okay, this is again parenchymatous tissue, and this xylem and phloem, when we study about secondary growth, they go in size, that is a different thing. For now, what you have to remember is that such is the arrangement, and such arrangement is known as radial vascular bundles. Now when we talk about radial vascular bundles, okay, do remember this conjunctive tissue as well. Moving further, these radial vascular bundles are also given another terminology, that whether they are exarch or endarch. Now this exarch and endarch arrangement is in terms of xylem. Okay, there are two types of xylem. Firstly, one is metaxylem, other is protoxylem. Now we are coming to different terms. If you have done them already, that is good. If you haven't done them, then again it is good, because you will get to know what it is now. One is metaxylem, other is protoxylem. Both of them are xylems, but at what stage of growth they are formed, on that basis we have two distinctions. Now as the name suggests, we know proto is something which comes first, okay, in the beginning. So the xylem that comes in the beginning is known as protoxylem. Okay, the xylem that is formed in later stages, or the newly formed xylem. Supposedly you have a plant that is 3 years old. So the xylem that would be formed in the first year of its growth, that would be protoxylem, and whatever xylem is formed now, that would be metaxylem. I hope I am clear with it. So we have two types of xylems. One is protoxylem, other is metaxylem. Now one thing more, how can you distinguish between protoxylem and metaxylem? By looking at the diagram itself. Now you see, when the plant is younger, or take your own case, whenever you are younger, your food requirement and water requirement is very less. Compare your water requirement with a small baby. So you would see that the small baby would be done with 2 glasses of water, and you will require 7 to 8 glasses of water per day. So by any case, if you were to put that requirement in a container, the container for the small baby would be smaller, and for you it would be greater. Same is in the case of plants. When plants grow in size, their water requirement is more. So the container through which the water would be conducted would be of a larger size. So you can make it out that the larger container would be formed in the later stages of the growth, as you can see over here. This particular xylem is having a greater radius and this is having a smaller radius. So you can make it from here that the one which is having smaller radius is protoxylem, while the one with larger radius is metaxylem. That is very easy to understand that when the plant grows in size, it is going to have greater water requirement. When it has greater water requirement, the particular part which is responsible for conducting water should also have greater radius. Otherwise, it will not be responsible, it would not be able, in fact, to conduct much water. So the lately formed xylem, or in the later stages, the xylem that is formed would have a greater radius, and that would be known as metaxylem. The one which is having smaller radius, as you can see, it is in the beginning, that is protoxylem, previously formed xylem, newly formed xylem. Now, in context to the arrangement of these two xylems, whether the protoxylem lies towards periphery or whether it lies towards the central part in the stele, we have two arrangements. You have to remember, one is exarch, other is endarch. Now, whenever this exarch and endarch reference is made, it is made with reference to protoxylem. Wherever the protoxylem is located, we always keep in mind where the protoxylem is located. So, if the protoxylem is located towards the endodermis, that means it is towards periphery of the stele, that arrangement is known as exarch. You know, exo means something towards outside, okay, and endo means towards inside. So if the protoxylem lies towards outside, it is known as exarch. So our vascular bundles in the case of Dicot root are exarch. Okay, in case it is opposite, these greater radii one circles are towards periphery and the smaller ones are inside, then that condition is endarch type of xylem. You have to remember these two things, you have to remember them, and by heart you have to keep in mind that this arrangement in the case of a Dicot root is exarch. We have exarch type of xylem which is present, in which we have metaxylem towards the pith and the protoxylem lies towards the endodermis. Please remember these things, okay? I hope I am clear that metaxylem is towards the pith, which is highly reduced, as I talked in the beginning, and protoxylem lies towards the endodermis. Protoxylem is having smaller radius. Now phloem is present. In between phloem there is conjunctive tissue, then there is pericycle that lies beneath the endodermis. Endodermis is having the Casparian strips. Few or many intercellular spaces are present in the cortex, and epiblema is having root hair. These things you have to keep in mind, okay? This is what you have to remember in case of the Dicot root. So I hope I am quite clear with it, that what are the parts, what are various arrangements and how it is well arranged. Now when we study the absorption of water, it would be very easy for you to understand. Before I end the topic, I just want you to get introduced to another term, that is polyarch. Now as I told, endarch, exarch, this has something to do with vascular bundles only. As you can see, in the case of a Dicot root, there are 4 pairs of vascular bundles: xylem, phloem, xylem, phloem, xylem, phloem, xylem, phloem. This is what you see. In case of monocot root, we are going to find polyarch condition. Now in polyarch condition, when there are 6 or more vascular bundles which are present, that condition is polyarch. Okay, now why I am telling you it over here, though it is not present, because as I told you in the beginning that the points of differences are a better way to learn, okay, what one is, what one basically is. It is better to understand from the points of differences. So the 2 points of differences between a Dicot root and a monocot root which you have to learn at Dicot roots level is, first is that the pith is highly reduced in the case of Dicot root, which would be different, okay, in the case of monocot root. And secondly, the Dicot root does not have polyarch condition. It is having 4, not more than that, 4 4 vascular bundles would be present, and in the case of monocot root we are going to see how many are there or if there are more. So this is all that you have to remember, keeping in mind what type of tissue is present in which part of the root, how they are arranged, and do keep in mind, because this is going to help you a lot in understanding the absorption of water by the root.